Welcome back to the second session of today's seminar with Jinwoo Leem from Alchemab Therapeutics, talking about deciphering the language of antibodies using self-supervised learning. Looking very much forward to it.
Great, fabulous. So can everyone see my screen?
Yes, perfect.
Excellent, great. Yeah, so I'm really excited to talk to you today about deciphering the language of antibodies using self-supervised learning. That is an incredibly loaded title, and we will go through little bits and pieces together. And this is a work that I've had the real pleasure of writing with the tech team guys here at Alchemab Therapeutics. So for those of you who haven't heard about Alchemab, Alchemab is now a small biotech company. So it's about 50 people big now, and we've recently had a $80 million Series A last year, and we mostly focus around neurodegeneration, oncology, and infectious disease. And the kind of premise of our company is to look for protective auto-antibodies from individuals and trying to see whether those antibodies could be repurposed as therapeutics. So the sort of gist of Alchemab's discovery approach is to look for resilient individuals. And what we mean by this is people who are perhaps healthier than expected. And among those individuals, we look for sort of protective antibodies that are common in between them. So those that are convergent. And then we use a target agnostic approach in order to understand what they are potentially binding to. If you want more details on what Alchemab does and just more about the company, then please feel free to reach out to me after the presentation. So I thought it might be worth just clarifying what is a B-cell receptor, just to make sure that everyone's on the same page. So B-cell receptors, as the name suggests, are proteins on the surfaces of B cells, and they're found in pretty much all vertebrates. And in its secreted form, a BCR is also called an antibody. So for the rest of this presentation, I'm just going to use the words B-cell receptor and antibody somewhat interchangeably. And it's very exceptional because they have a huge level of diversity, as I hope many of you are aware. And what makes antibodies, for me, very interesting is the fact that they could bind almost any molecule because they have extremely high sequence and structural diversity. And once it sticks onto something, it activates other cells of the immune system and helps to clear that target away. In particular, antibodies are characterized by 6 so-called CDR loops, and they're the colored bits in this sort of bottom structure. And in particular, the thing in the middle, which is your CDRH3, is the most important. And typically, that's where most analyses and kind of effort is focused on understanding. And I've mentioned that B-cell receptors are proteins. And what's been kind of interesting lately is to look at B-cell receptors as text data. So we know that proteins like B-cell receptors are actually just a string of amino acids. There are these linear polymers that you can think of as just this chain of amino acids. And for each and every one of these individual amino acids, you could represent them as individual characters of our human language as the alphabet. And in fact, this sequence alone should give you pretty much all the information that you need in order to understand the structure and thus the function of that antibody. And we see this kind of for pretty much every protein in the universe, and it's not just for B-cell receptors, but just to show as a case in point that you can, in theory, represent this 3-dimensional sort of thing into a linear kind of text representation to kind of understand and see in bulk as well. And what makes B-cell receptors so special? So in addition to the fact that they could recognize pretty much any molecule in theory, it's the fact that they are unlike anything else in the protein universe. So the CDRH3 bit, which I mentioned earlier, has a huge range of length. So it can be anywhere from 5 to over 30 residues long. And none of them really have any kind of common motif that makes you go, oh yeah, well, you know, definitely it's going to do this because it has this motif. It's kind of difficult to make that out. And you can tell structurally that each and every CDRH3 has very different shapes, despite the fact that the rest of the antibody structure is more or less pretty consistent. The other sort of, well, however you want to interpret it, what's curious is the fact that some antibodies, despite having very sequence dissimilar kind of properties, like these two B-cell receptors, even though their sequences are very different, they can bind effectively the same antigen and in the same epitope as well. And so we have this unusual sort of conundrum where you have this huge diversity. And on top of that, what makes it even more fascinating and a bit more frustrating in many ways is the fact that you can't really kind of predict the antigen from sequence alone. And of course, in relation to the B-cell receptors sort of huge level of theoretical diversity, the number of labeled data sets, so what I mean is a mapped relationship between a B-cell receptor sequence to its antigen, is actually quite sparse. So even though now there are public databases like the OAS, where you have sort of in the order of 1.5 billion sequences that are available in the public domain, you actually only have maybe in the order of several hundreds, if not several thousands of B-cell receptors where you know kind of what they bind. And so what we really want to get to is, how do we have a better understanding of the sequence space? Could we even get there? Could we even understand what B-cell receptors do? And once you do that, then you're able to actually have a better understanding of the immune response, because then you could then start making kind of inferential judgments about, okay, well, this B-cell receptor is predicted to bind this thing, and therefore it'll have this kind of effect on your immune system. And so what we want to do is to capture the sort of quote unquote, the semantics of that B-cell receptor sequence. And the best way to do that, we think, is to use machines, because machines can process loads and loads of data and make good inferences, at least better than humans. And so in order for us to input text data like B-cell receptor sequences into a machine, we have to convert it into some numeric format. Tao's talk kind of touched on this already, so I think we now have a slightly more familiar idea about this concept. So essentially what we want to do is a form of representation learning. So how do we go from sequence into numbers? Classically, there's been different ways of doing this. So we can use, for example, k-mers. So a certain Victor Greif, for example, has shown that you could look at k-mers within your CDR sequence. You can look at the number of times that a particular k-mer, or sort of like a subunit of that sequence, appears in your CDR, count the number of times, and apply, say, another machine learning method on top. You could also use something called position-specific substitution matrices, which is quite old school, but essentially what it does is to look at each and every position in your CDR sequence. So as columns, and you look at which amino acids are present in each and every one of those positions and look at the probability distributions. The other is to look at physicochemical features. So Tao's talk kind of mentioned this already. So you can think about how certain amino acids are, say, polar or hydrophobic or whatever have you. And for each and every one of those physicochemical properties, you have a set of numbers to represent your amino acid sequence. However, what our work focuses on is to use machine learning to do this part as well. So it's to use a machine learning model that converts this sequence into some vector of numbers, some series of numbers, which should have some sort of capturing ability to represent that amino acid sequence into some numeric space. There's lots of work going on, especially in the last year alone. And our work is just one of the many things that are just kind of happening in this space. And in order to do this sort of representation learning, going from sequence to numbers, one of the best ways to do this is to use a set of machine learning models called deep learning models. And deep learning, for those of you who are not so familiar, deep learning is basically a set of, it kind of refers to a set of machine learning methods that are really good at finding hidden patterns in large data sets. And the reason why now we have good, say, image recognition methods is because they've seen loads and loads of images of cats or dogs, and it figures out, okay, well, what makes a cat, and then figures them out. Similar way, we can do that with text data and with BCR sequences. Specifically, the machine learning model that's really been kind of making waves in this space is transformers. And transformers are a sort of set of models that have made waves in natural language processing. So starting from 2017, there's a paper from Google titled "Attention is All You Need," and they kind of introduce this transformer model. And later in 2018, the BERT transformer kind of comes to the scene and says, oh, hey, if you give transformers loads and loads of unlabeled text data, then it turns out it can then be transfer learned. So what we mean here is after it's learned something about the text, it can do other inference tasks like detecting, say, nouns or verbs, or detecting if a sentence is swearing or it represents something happy or whatever, and it does it really well. So then come about sort of 2020, people then start to go, hey, this transformer thing's really cool. Why don't we apply for proteins? So then this is when you start to see the emergence of protein-specific transformer models like ESM and ProBERT. And in 2021, in particular, we now see transformers being incredibly mainstream, and it's now used for BCRs. So I should say that there are other really fantastic pieces of work in the space, like the BioPhi transformer Rs, AntiBERTa, AntiBERTa, and Apply. And I highly recommend reading all those papers if you have time, because they're really well written, really thorough as well. But how do these things learn? Like, how do they then have this kind of magical ability to have these astonishing results, as they say? So here, this is where transformers learn by an approach called self-supervised learning. So what does that mean? So here, how transformers learn is if you have a sentence like, I like eating chocolate, and I do like eating chocolate. But anyway, if you have a sentence like, I like eating chocolate, then what we do is to randomly mask out random parts of the sentence. So you might mask out the verb eating. And then so you have, I like blank chocolate. Based on the context of the sentence before and after the word, it tries to work out which verb should fit in that blank. This entire task is called masked language modeling, and it falls under the umbrella of methods that one can use for self-supervised learning. But I would say that this masked language modeling is probably the most common for transformers. And in a similar way, you can do the same thing for amino acid sequences. You could randomly blank out one position, and then given the context before and after, you try to work out what amino acid fits best in that blank. And so the real kind of key element here is that transformer models learn sequence context. It doesn't just learn, here's a distribution of amino acids. It learns a distribution given the context of everything around it. And once you pre-train transformers using this sort of masked language modeling task, then what transformers do is to effectively generate a set of numbers for each and every one of those sequences. So it'll look at a sequence, generate a number, look at a sequence, generate a number. And then with these numbers, then you could do lots of interesting things. So here you can do, for example, like dimensionality reduction. So here, this is a figure from the ProTrans paper where they've essentially applied dimensionality reduction, like Tisney, on these sets of numbers and plotted each protein sequence as a dot. And you could quickly tell that, okay, there are some sets of numbers that correspond to eukaryotic proteins, some sets of numbers that correspond to bacterial, et cetera, et cetera. And in the same way, you could use those sets of numbers as input to another machine learning model. And this part is called transfer learning. So what you do here is to use a second machine learning model that takes this as an input and makes new predictions. So here, this is a figure from the tape transformer paper where they take input amino acid sequences, put it through, get an embedding, and then try to predict whether it's a helical region or a coiled region, etc. So then how does AntiBERTa work? So AntiBERTa is yet another kind of transformer model, and it's pre-trained by looking at 57 million human B-cell receptors. So what we do here is we get loads and loads of sequences from OAS, and we put it through a transformer model, and the transformer model then, as I explained before, randomly blanks out bits and pieces, tries to fill in the blanks, and then once it fills in the blanks, it gets an understanding of that B-cell receptor sequence, and then generates 768 numbers. So for each and every B-cell receptor, the key message here is you get 768 numbers. Once you have those 768 numbers, then you could start running it for downstream tasks, and you do it on sort of smaller labeled data sets, such as paratope prediction, which I'll describe in a couple of slides' time. And in order to do this, we used 8 NVIDIA GPUs for about 200,000 steps. So it's not the longest training regime in the world, but it was sufficient enough, in our view, to kind of get this representation learning going. And what's interesting is that our model is currently the deepest BCR-specific transformer model, and we're planning to train larger models and train it for longer periods, and think about using other pieces of the OAS to kind of have a more fuller kind of picture of the B-cell receptor space. And what we found really curious is the fact that the embeddings that we got out of our AntiBERTa model are able to show zero-shot learning performance. What we mean by zero-shot learning is the fact that we didn't give any information really upfront, yet the transformer was able to pick out functional aspects of the BCR. So what this plot is showing here is, this is effectively the same UMAP plot for embeddings from a BCR-specific transformer or a general protein transformer like AntiBERT. Here in this panel, what we have is for each and every dot, which represents a B-cell receptor, we're then coloring it by, for example, the immunoglobulin V gene family. And we can see that, the BCRs largely separate by whether it's, say, V4, V3, V2, etc. You can maybe argue that the AntiBERT one is doing this slightly better, but the general gist is that sequence families do split out. And it's interesting because we don't necessarily tell the model, hey, this particular BCR is, I don't know, a V434. We don't really tell it that. But just by looking at the text sequence, it already figures it out that actually, these things look similar. Next, we then colored the points by the number of mutations that are in the BCR sequence. And again, we don't necessarily tell the transformer model, hey, this BCR has, say, 20 mutations and this one has 10. We don't tell it that. But even then, it kind of magically works out. Hey, the BCRs on the left-hand side of the plot look less mutated than the ones on the right. And we can then start to see some separation in the BCR-specific transformer compared to general protein one. So the BCR-specific one, you can clearly tell a sort of sense of directionality going from left to right, where you're getting more and more mutated sequences, whereas with AntiBERT, it's not entirely clear that that's the case. And finally, this is kind of my favorite one out of the lot. It's showing how we've colored the BCRs dependent on their B cell provenance. So whether they were from naive B cells in blue or memory B cells in pink. And again, we didn't tell the transformer that each of these BCRs are from, say, naive B cells or memory B cells, but it sort of naturally separates them out, that we can see a separation even though we don't really tell the model up front, whereas AntiBERT doesn't really do that at all. So what's really exciting about this is the fact that perhaps our transformer is capturing some functional aspects of the BCR sequence, and maybe some that we currently don't understand right now, but this is where the potential lies. And I think this is incredibly fascinating that the model is able to do this without us providing this information explicitly. Another key part of transformers is the fact that transformers have what's called the self-attention module. And the self-attention module helps us to have a better understanding and interpretation of what the transformer is learning under the hood. So the previous image kind of showed, hey, it does this magical thing of learning function, but how does it do that under the hood? That is being done by a component called self-attention. So self-attention, in a nutshell, tries to work out how different words are related to each other within a larger sentence. So if we go back to the sentence, I like eating chocolate, then you could reasonably infer that the word eating is probably very important for the next word, chocolate, and maybe for the subject I, but perhaps not as relevant for like, but you could quickly work out the sort of dependencies and importances between the different words. And similarly for self-attention, you can do that for the individual amino acids and how they're related to each other. And what we find is that AntiBERTa's self-attention module, it's related to structural contacts that are observed in antibody structures, kind of like what other people have seen before with protein transformers. And so we can use AntiBERTa not only to figure out, functional aspects of the BCR, but also potentially structural aspects where we can quickly work out, hey, maybe, these positions are really close to each other and form, interesting interactions, say. And once we have pre-trained this transformer, then we decided to apply for a particular downstream task, which is paratope prediction. So the gist of the paratope prediction task is, if we have a known antibody antigen structure, say in the case of pembrolizumab, where we know which positions of the antibody are binding to the PD1 antigen, then we try to tell the transformer, now that it's learned something about B-cell receptors, can you now try to tell me which position along this entire VH will be the bits that bind the PD1 antigen? And these are actual predictions from AntiBERTa for pembrolizumab. And you can tell that it's fairly close. And here we find that AntiBERTa does achieve sort of state-of-the-art prediction results in comparison to already sort of publicly available tools. And we have pretty good precision recall and AUC. I won't go through those kinds of numbers. In particular, you can see that in our preprints. But perhaps what's really interesting is the fact that, for example, for some antibodies, like this one, which is the light chain of tralokinumab, where it uses amino acids outside of its CDRs, like this histidine here in framework, and the NSG motif here in the framework again, and this aspartate here. AntiBERTa is able to pick out these non-CDR positions as contacts, here shown in the pink. So even though it doesn't get all of them right, at least what's really interesting is the fact that it can pick out these non-CDR positions that can be really important for binding. So going forward, I mean, what's next? Like, what can transformers do? What kind of work, can we really explore? I think AntiBERTa is just one of the many papers that shows how transformers are incredibly flexible, and it can show really good performance both in zero-shot and transfer learning paradigms. And I won't go through the details. They are in our preprint, but I think it's an exciting time to work on transformers in this space. And it's really interesting how the different papers I mentioned, all saw transformers as a platform to do things like humanization, or affinity maturation, or kind of antibody design, or error correction. And again, it's really finding the overlap between, kind of lab use case and kind of how you codify that. And once you find a really sweet intersection, I think, transformers can really, no pun intended, be transformational in, how this field evolves. And I would say that, transformers have been much more commonplace in the small molecule kind of universe. And I really do see similar advances kind of happening in B-cell receptors and sort of protein design also as a whole. So in summary, I wanted to say that B-cell receptors are exceptionally diverse, and they have a huge level of structural diversity that's unlike any other protein in biology. And because of that diversity, we want to use deep learning in order to understand B-cell receptors. AntiBERTa is one of the many transformers that are out there now in the public domain. And it's tied with Applying as the deepest kind of BCR-specific transformer model. And we showed how it learns functional aspects of the BCR, whether that's, B-cell provenance or, paratope positions, et cetera. And especially important is the fact that it can do this without necessarily having some of these things known up front. And as I said, it's an incredibly hot field, and there's lots of other good papers in the space, so do have a read. And yeah, I think it'll just go as far as your imagination. So yeah, please feel free to drop me any questions or reach out to any of us. And I just want to give a quick shout out that we do have openings for one bioinformatician and one machine learning scientist. Thank you very much.
Thanks so much for the very nice talk. Are there any questions? Maybe I will start with the first question. So can you show slide 11 again?
Wow, that's incredibly good memory.
No, it was written down. So
yeah.
So what would you say to devil's advocates if one would ask you, so here for the clustering, right? You say it's clustering by function. If you just use sequence similarity, how would this clustering look like? And isn't it a bit obvious that you cluster a bit by V genes because they are different per se, for example, or that you cluster by mutations or not, but because they're mutated and they're non-mutated, and they're all also a bit confounded, right? So you have memory B cells are mutated, naive B cells are not mutated. So these labels are not orthogonal to one another. So what would you say to that? Yeah.
Oh, well, thank you, Devil. So a couple of points. I think sequence similarity is a really interesting one. So I think one could maybe look at doing like sequence similarity and then doing like an MDS and then comparing how that projection compares to this one. I must admit, we haven't tried. That would be the number one thing to do. As for this, I'll spin it the other way. If I didn't see this, if I didn't see the V gene split and the mutation split, I would almost ask, then what the hell have I done? So in a way, like these first two plots are sort of a good sanity check for me to say, okay, right, it does do something that I would reasonably expect as a prior. And again, even this like memory kind of naive B cell split, it isn't 100% like accurate. And, like you say, they're not orthogonal, right? Like they are highly correlated labels. That's absolutely true. But I think even then we see, for example, even in this cluster, we see some sequences that are actually not all that mutated, right? But even then, they're the memory B cells that it picked out. So those are the ones that I'm most interested in.
Yeah, I think it's just nice, to the people that, are a bit, doubtful of machine learning, to give them a bit of a stronger, like why is it really useful, like give us something, a really killer application or something. Yeah,
definitely.
Which might be the paratope thingy and the epitope and so on.
Yeah, definitely. I think for any of these things, I think it's always nice to have some quote unquote fallback applications where you and the experimentalists both know, like, right, if a machine learning model works, this baseline task, which should be, should be the thing it does, should work.
Sure. I agree. Any questions? Any other questions? If not, I have lots of questions. So
I might have scared everyone off. I don't know.
So there is a question in the chat. And then there's also a raised hand. So yeah, we will first.
I have one question. If you look at the memory cells on the right-hand side cluster, the red cluster, the pink cluster, I see two subclusters. Do you know what is the difference between them?
So that's probably to do with one of them being V4 and one of them being V3. Yeah.
Okay.
I actually don't know what order to do these in, but I'll look at the chat first.
Yeah, chat and then the raised hands. Yeah.
Great. Okay. Yeah. Oh, very good question. How many trials did we need? Lots. is the short answer. But I think the other as well is I'm kind of finding this myself, that a lot of this also has to do with experience with this field, and just having a kind of flavor for what the different hyperparameters do for transformers. So I'm afraid it's going to take a lot. But yeah, hope that helps, Peter. So I think Gisela, am I saying that right?
Yeah, that's correct. Thanks. So I had a question regarding the training and fine-tuning on smaller data sets. It is really interesting. I was wondering if you have tried already for some projects, and how small can a small data set be there for fine-tuning?
Yeah, That's a very good question. I think that's that's the equivalent of how long is a piece of string kind of question, right? So I think we haven't really tried on very small data sets, but it might be worth investigating. But it might also be down to the nature of the problem itself, right? So short answer, I don't know what's too small or big enough, so to speak, but it might be worth looking at different kind of fine-tuning tasks and then kind of benchmarking it from there, if that helps.
Yeah, thanks.
Right.
And Yuta is the next one. Yeah.
Hello, thank you for the talk. So in order to produce these vector embeddings of these B-cell receptors using BERT, you would have had to go from, a sequence of BERT embeddings of the individual amino acids somehow to reduce it into one fixed size vector, right? So how did you do that?
Oh yeah, really good question. So we just took the average. It's a really good question. So basically you have a three-dimensional tensor. So you have a tensor that's n sequences by L length, which is say 100 something residues and 768 numbers. You just collapse them so as to take the average along the, I always screw this up, but basically along the length dimension so that you get n sequences, 768 numbers.
No, yeah, thank you. That makes sense. And so when deciding to do the averaged collapsed vector as the fixed size embedding, I know some work with BERT in the natural language field, they make like kind of specialized training regimes that incorporate the assumption that the model will eventually be used by creating averaged embeddings and stuff like that. Did you do a particular kind of training regime with that assumption, or did you just train based on masked language or masked amino acid modeling, I guess, only?
Yeah, so we just used masked amino acid modeling for now. We didn't really have any particular aims, as it were.
Okay, I see. Thank you.
Nice, thanks.
So we will have two more questions and then we will end. So Habib, and then the last question.
Hello, thank you for the talk. If you don't mind, can you go back to the slide with the multiple transformers and followed by, yes, this one. Can you just repeat how SAPDAB here is participating?
Yeah, that's a really good question. I sort of glossed over that detail. Apologies. So here, basically, up to this point is really the transformer part. What the SAPDAB component here is saying is, okay, now that the transformer has done some form of learning about the sequence itself using masked language modeling, can I then use a smaller data set like SAPDAB, which is much smaller than OAS, but it'll have interesting labels, like what antigen it binds to, what are the paratope positions. So then to sort of fine tune or specialize the transformer now to then do this sort of prediction task based on stuff that's in SAPDAB. That's the idea.
Do you think that can limit the output because SAPDAB dataset is quite limited or small?
I'm not sure about what you mean by limiting output, but
there's so many antigens that the SAPDAB antibodies can bind to. So by specifically training on the SAPDAB dataset, would you be limiting or restricting the training?
Yeah, I see what you mean. So one of the reasons why we used SAPDAB is because we were especially interested in paratope prediction, and to get paratope kind of annotations, you really need structural information, unless they somehow worked out otherwise. But typically, you'll need structural data, and that's why we kind of stuck to using SAPDAB in this case.
Okay, thank you.
Okay, so then the last question. Yeah. Please.
I have a question. So I think there are two choices to what to learn from the BCR sequence, and I want to know, is there any background in choosing an amino acid sequence rather than a nucleotide sequence?
Yeah, it's a great question. So I think with nucleotides, it's going to be a bit more of a challenge for two reasons. So one is that nucleotide sequences are longer. So that means that your learning process has to deal with bigger tensors. And typically that just means it's a little even slower than, and it'll take more time than using amino acid sequences. And the other is that with amino acids, for every amino acid, you've got sort of 20 positions. So in a way, you have more sort of token space to spread your information. Whereas with nucleotides, you've only got 4 choices. And so my expectation is that the learning regime will just be much tougher with an even more reduced alphabet. So typically transformers actually work with anywhere between sort of, I'm going to say about 30,000 words or so, right, in its vocabulary. But for proteins, you've only got 20 words in your vocabulary. So you've got to somehow condense a lot of information into just 20 words, which is pretty crazy, right? It's like, can you imagine a language where you only have 20 words? That's going to be pretty hard, right? So there's a lot of information compression to do. So I think with nucleotides, this problem becomes even more like this, it just becomes worse. You just have to do it in 4 words rather than 20. So it just makes more sense to do it on amino acid sequences.
Okay, thank you.
Great. So with this, we have come to the end of the seminar. Thanks again so much for your great talk and all the questions. And we will be back March 24th in 1 month. Thanks so much, everyone, and have a good day.